We focus a lot on glucose in metabolism. This is for a variety of reasons, including that it really is at the core of a lot of biochemical metabolism. We've talked a lot about glycolysis, that is when we break down glucose catabolically to basically generate ATP. So in glycolysis, we take glucose and we break it down into pyruvate. Later, we'll follow back up on that pyruvate's fate. We'll see it go through the citric acid cycle, oxidize it even further, generate more electrons that we can take through to the mitochondria, go through the electron transport chain, oxidative phosphorylation, and get ATP. So that's all great, and we'll return to that catabolic story soon, but glucose has other fates. One of these fates that we'll talk about later in the semester is the pentose phosphate pathway. So this branch over here. This pathway is really great if you want NADPH, or if you want ribonucleotides, if you want deoxyribonucleotides, if you want your various cofactors. So the pentose phosphate pathway is going to be great for that. There's also a pathway that we won't have time to get into, but it's called the hexosamine pathway. Basically, here you branch off at fructose 6-phosphate, and you end up making those GalNAcs, so those modified monosaccharides that you can use to make more complicated glycans. So glucose is great for a lot of different things, and we can get it from a lot of different places. We can get it directly from our diet. We can get it from the interconversion of some of those other monosaccharides. And we can get it from the breakdown of, say, disaccharides like our sucrose, that's just glucose and fructose, or our lactose, that's glucose and galactose, or our maltose, that's just 2 glucoses. If you break up these disaccharides, you get 2 monosaccharides, which can then enter the pathway. So either directly as glucose, or they can get converted into glucose, and all of those can then feed into this central glucose pool. So glucose is really great, but we wouldn't want to store just individual glucose units. So yes, it might be easier if you need a quick source of energy. Oh, we've just got some glucose already free floating. But if you've got a bunch of free floating glucose, then what's going to happen is you are going to have hypertonic cells. So there's too much stuff inside of your cells compared to how much stuff is outside of your cells. The water's going to rush into your cells are going to burst, and not good. Thanks, osmosis. But there's a solution. And the solution is that instead of storing individual glucose monomers, we can link them up. We can form this big complex of these glucose units in the form of glycogen. So glycogen, remember, is going to be our storage carbohydrate. So our storage polysaccharide. So in plants, remember, they use starch. We use glycogen. So what is glycogen? Glycogen is basically this protein in the center called glycogenin. And then it has all these branches of your glucose. So we have those 1,4 linkages, which will give us our straight chains, and then our 1,6 linkages, which will give us our branches. So if you need to review your carbohydrate your carbohydrate biochemistry, go back and look at those structures and things and see how we have those 1,4 and 1,6 linkages. Why all these weird linkages? Well, the branching makes it so that we have multiple places that we can kind of bite off from. If we want glucose as a source of energy, either to use for ourselves, so our liver cells, our muscle cells we see, they're going to be like really selfish, so they'll break down glycogen and they'll use the glucose for themselves. Our liver, on the other hand, well, our liver's job, well, one of its job is to regulate our blood sugar. And so if the blood sugar is high, the liver will take it in, will send out, like the pancreas sends out insulin. We'll talk more about this, but that gets the liver to take in the glucose and branch it up into that glycogen, build it up into that glycogen. And if it's low, then glucagon gets released and by the pancreas, and that gets the liver to break down glycogen and ship out the glucose. So unlike the muscle that's using it for itself, the liver is going to ship out that glucose, and that's going to help bring blood glucose levels back to normal. So we want to be able to kind of quickly build and break down this glycogen, and the branching is going to allow us to do that. If you think about it kind of like at a grocery store, how if you have the same amount of people, but there's only one cash register open, you're going to have one long line, and it's going to take a lot of time to get through everybody. However, if you open more lanes, well, now you can assist the people more quickly, and your lines will move faster. Similarly, by having these multiple branch points, we have these different places where you can have this breaking off occur. We'll talk later about how this breaking off happens through a couple of different enzymes. So we have our glycogen phosphorylase, which can do the straight chains, and our debranching enzymes. So our branching enzymes and our debranching enzymes, these are basically like opening up new lanes at the supermarket, or transferring lanes. So when one lane closes, they've got to move everybody to the single line, and then if another lane opens, then they move some people from one line to another. That's kind of like our branching and our debranching enzymes. We're not going to go too much into the details of the specific enzymes in the pathway in terms of the chemical reactions, but we will talk a lot about their regulation, and I'll go through what I would like you to focus on. But let's get back to our story and think about what is the big picture of our glycogen metabolism. So we have this story that we have this glucose, and we have these different fates, and we're going to focus on over here, this glycogen metabolism side of things, where we take this glucose, we're going to use this enzyme called phosphoglucomutase to convert it to glucose 1-phosphate. We're then going to activate glucose 1-phosphate into a form that we can add through this UDP glucose pyrophosphorylase. Then we're going to add those UTP glucoses together to form glycogen using this glycogen synthase. And then we can reverse the process, break off glycogen units to give us glucose 1-phosphate. That's going to be by our glycogen phosphorylase. Don't worry about if this seems overwhelming right now. We're going to go step by step. But I do warn you that this is going to be one of the more complex pathways we're going to talk about, especially when it comes to regulation. This is probably the most complicated regulation we're going to see. And so the good news is that if you can understand this, then you're going to have an easier time understanding the rest of the regulation that we talk about. And it's really great because it's going to show us a lot of examples of the different types of regulation that we'll see in metabolism. So we're going to see both covalent and allosteric regulation. So we're going to see covalent regulation. So this is like via phosphorylation and dephosphorylation, by kinases and phosphatases that are going to be regulated hormonally. So we'll see that our main hormonal regulators of our glycogen synthesis and breakdown are going to be insulin, which is going to say, well, okay, insulin, we've got a lot of blood sugar, let's put it together, let's store it. So let's promote glycogenesis, so the making of glycogen, and let's inhibit glycogenolysis, that is the lysis or breakdown of glycogen. On the other side of things, if we don't have much glucose, we want our liver to ship out glucose, then we're going to want to have glucagon that's going to then activate glycogenolysis, so activate the breakdown, and inhibit glycogenesis, so inhibit the storage, because we want to break it down and ship it out. We'll also see how we can have regulation by allosteric molecules. So these are small molecules, things like we'll see glucose and ATP and ADP and AMP. These indicate nutrient availability and energy status, and are going to directly activate or inactivate various enzymes, or make it easier for them to be activated or inactivated by our kinases and phosphatases. I know, it's complicated, but we'll get through it together. One of the tools that's going to help us get through it together is going to be our metabolic spreadsheets. So what I want you to do is to put all of the enzymes, these core enzymes we're going to talk about, into your metabolic spreadsheet. So each of the enzymes should get a row in the enzyme name and abbreviation column, and then you can fill these out as we go along. Remember that you might not have all the information for all of the columns yet, and some of the columns might not really be applicable, but do your best to fill it in, and this will help serve as a study guide. These are the key enzymes of glycogen metabolism. When it comes to the actual metabolic enzymes, the enzymes that are going to go in the carbohydrate metabolism tab of your spreadsheet. These enzymes are, there's the one that controls the entry and out of pathway into and out of the entry into and out of the pathway. This is going to be our phosphoglucomutase. It takes glucose 6-phosphate, which remember is what we get from glycolysis, That's our hexokinase or our glucose kinase step. It adds that phosphate to the 6th position, and that traps things in the cell. That prevents it from getting out of the cell because we don't have those glucose 6-phosphate transporters. And it also is going to help kind of basically put a little energy investment in, so we have a little energy down the line. It keeps all of these components phosphorylated so they can't escape either. That phosphorylation is really great. We're going to talk a lot more about glycogen, about glucose metabolism, the regulation of glycolysis later. But we talked about a little about how this F16BP or this this PFK1 step, this PFK1 step is the major step of regulation because this is kind of like the committed step of glycolysis. So if we looked at where we branch for the pentose phosphate pathway, it's up here. Where we branch from glycogen metabolism, it's up here too. So it's this glucose 6-phosphate that's really going to be kind of like the starting point for a lot of pathways. Also, this fructose 6-phosphate, this is where we went off for the hexosamine pathway. So all of this is before we actually do this PFK1 step, we add that 2nd phosphate. At this point, our glucose has a lot of different fates. And so this is why we consider the PFK1 step to be really the committed step of glycolysis, and why we'll see that we regulate it so heavily. But going back to our story, we have this glucose 6-phosphate. We can interconvert it readily using phosphoglucomutase to move that phosphate to the 1 position. So that's going to be the role of our phosphoglucomutase. Okay, so that was that enzyme. We'll also see, we'll go through these. As in glycogenesis, we have the UDP glucose pyrophosphorylase that's going to basically take that glucose and make it UDP glucose. It's going to activate it so it can be added. It gets added by this glycogen synthase. Then remember, if we want to branch things, we need this branching enzyme. Glycogenin, that's going to be that protein at the heart of the glycogen, which actually is an enzyme that helps start the process, as we'll see. In terms of the breakdown, this is going to be done by our glycogen phosphorylase. So don't confuse phosphorylase with a phosphatase. A phosphatase removes a phosphate group, and a phosphorylase uses an inorganic phosphate to hydrolyze something. And we'll get into what that looks like in a minute. And then we need the debranching enzyme in order to break things off. So you should go ahead and pause the video and put in a row for each of these enzymes in your carbohydrate metabolism spreadsheet. Dive in and look at a little more detail. Remember to keep pausing and filling things in as you go. It'll make things a lot easier, and then you'll have this source that you can look back on when you're trying to think through things more details. So going back to our big picture idea, we have our glucose. We convert it to glucose 6-phosphate through our hexokinase, or remember in our liver, that's called glucokinase. That's going to trap the glucose in the cell, keep it from getting out, make it so that when we break apart the glucose into different pieces, those pieces are going to be phosphorylated and they can't go away. They've got a little energy in them so that they can be activated for various processes. So that phosphorylation step is great. But when we want to link up the glucose, we're going to want to move that phosphate to the 1 position. We do this through phosphoglucomutase. We then activate it to UDP glucose using this UDP glucose pyrophosphorylase. Then we're going to hook up those UDP glucoses using glycogen synthase. If we want some branches, we use this branching enzyme. We get our glycogen, we break down our glycogen. If we want to debranch it, we've got to use the debranching enzyme. We're going to then use glycogen phosphorylase to break off little pieces. And then those pieces are broken off as glucose 1-phosphate that we can then go convert back to glucose 6-phosphate and do whatever the heck we want to do with it. We're not going to worry too much about the various enzymatic reactions, but I will just take you really quickly through them. Focus more, though, on the regulation. So this step where we basically activate the glucose. So we're not adding them as individual glucose units. We need to first pre-invest some energy. So we're going to add them as what we call sugar nucleotides. We already saw some sugar nucleotides. So like our GDP mannose and stuff like that, those are sugar nucleotides. Basically, you have a sugar attached to a nucleotide. It's kind of like prepaying the cost for linking them together. Basically, we're going to link together these UDP glucoses, and we're going to make those UDP glucoses from a glucose 1-phosphate, not glucose 6-phosphate, but phosphoglucomutase is going to readily interconvert those between one another. So this is going to give us our way into and out of the pathway. So we started with our glucose 1-phosphate. So we had one phosphate group. We moved it to the position that we wanted it, but now we're going to add another phosphate group and our ribose base. So in this case, it's going to be uridine. When we do this, basically the way we add it is going to release this inorganic, it's going to release this pyrophosphate. If you remember back from when we talked about kind of like ATP hydrolysis and stuff, if we have this inorganic pyrophosphatase that can basically break this pyrophosphate into two individual phosphates. They've got much better resonance. They need that repulsion. This is going to be a highly exergonic reaction, a very, very favorable reaction. This is going to help drive the production of our sugar nucleotide, in this case, our UDP glucose, which is then ready to be added into different chains. So now we've got our UDP glucose and we're ready to piece them together. A slight challenge comes from the fact that our glycogen synthase can't actually start a chain. Kind of like how in PCR, our polymerase couldn't just start from nothing. Our glycogen synthase can't start from scratch either. So we need to add a primer, not a DNA primer here, but just a short strand of our UDP glucose units. So a kind of mini part of our glycogen, the beginning of the chain. The beginning of the chain is actually added by that glycogenin protein at the very center of the chain. So this glycogenin, it serves as kind of like the central hub, but it's also an enzyme. And so it can add a short chain to itself and make a short chain on itself that then can be added onto. So this glycogenin protein is really cool. I remember I was still so shocked when I learned that like glycogen had this protein at the heart of it, this glycogenin, but it's really pretty awesome. And so you can go and look at like actual images. And so it's a ton of these glucose units and this big branching structure, but then this super powerful protein at its heart that really gives it a place to start. So that's our glycogenin. And then we'll move on to our glycogen synthase. And then if we want the branches, our branching enzyme. A note about terminology. So they're going to you'll see a lot of reference to reducing and non-reducing ends. If you need to go review this. But basically a reducing sugar has like an aldehyde and a reducing end has an accessible aldehyde group. So basically it has a group that can get reduced, that can reduce something that can get oxidized. And so we can oxidize an aldehyde to a carboxylic acid. It may not look like, if you look at it in the ring form, it doesn't really look like you've got a reducing end, but the reducing end basically is, if you think about the glucose being able to open up, if it opens up, then you're going to have an accessible aldehyde group that then can serve as a reducing sugar. So when we talk about a sugar with like a free anomeric end, that's going to be our reducing end, and our non-reducing end is the other end. So our reducing end has the anomeric carbon, and then the other end is the non-reducing end. And it's this non-reducing end that you're going to add the different units onto. So now we have our UDP glucose, and we're going to add it to the non-reducing end of our growing chain. So we can't add to the end that's already formed a linkage, but we can add to this kind of free end. This is going to be our non-reducing end. We use the reducing end to form one of these linkages already. So our glycogen synthase can make these straight chains, but then if we want to branch, we're going to need our branching enzyme. So remember the straight chain, that's our alpha-1,4 linkages. The branch points are going to have our alpha-1,6 linkages. This is like opening up a new lane of the supermarket. Just like you need a new cashier, you need this glycogen branching enzyme to open up that lane. And so that's what we see here. Now we've got our glycogen, all's going good, but now we want to break some down. What might signal that we want to break some down? All right, something like glucagon. If we need that to release more blood sugar, that if we want to release more glucose to raise our blood sugar levels, we're going to want to break down glycogen. We're going to want to do glycogenolysis. So we're going to need the branching enzyme to remove those branches and glycogen phosphorylase to break things off. So remember, not a phosphatase. We're not removing phosphate groups. It's a phosphorylase. We're actually going to be adding a phosphate group because we're using inorganic phosphate to break off the chain. When we do this, we add that phosphate group. So we're releasing a unit as glucose 1-phosphate, and we're releasing it from that non-reducing end. So the same end that we added onto is the end that we're going to break off of. like that plane. The first person in is going to be the last out, the last in is the first out, except that things are a little more complicated because you've got to move over the branches. Don't worry about the details of how all this happens, but basically this is going to happen with this debranching enzyme. And one of the consequences of this debranching enzyme, however, is that when it releases, it's going to leave a free glucose that just gets released as is, rather than as one of those glucose 1-phosphates. Why do I mention this? Well, if we have that glucose 1-phosphate, we can readily interconvert it to glucose 6-phosphate. And therefore, we don't have to put in the energy, put in the ATP of this first step of glycolysis to actually this hexokinase step where we make the glucose 6-phosphate, because we're just converting from glucose 1-phosphate. We already paid for it. And so now if we were to go through and we were to do glycolysis, we would only have to invest 1 ATP and we would make 4 ATP. So on net, we would generate 3 ATP per glucose from our glycolysis if we start from glycogen. But we did have to put in the energy to begin with. So there is that. Okay, but that's going to be like in your muscle cells if they're actually wanting to take that through glycolysis. But then remember, like your liver, well, its job is to ship out the glucose. So when it's breaking down glycogen, it's typically to provide it to your body. And so there, instead of actually going and breaking down the glucose, you're going to want to go in the gluconeogenic direction where you use that glucose 6-phosphatase in order to release the phosphate group to get free glucose that you can then ship out. Now, just like we talked about how we wanted to kind of reciprocally regulate things so we didn't break things down while we were making them and make them while we were breaking them down, similarly here, we don't basically want to be taking that glucose 6-phosphate and converting it into glucose in the cytosol that could then be taken and rephosphorylated to form our glucose 6-phosphate and enter kind of like a vicious cycle. Instead, what we're going to do is we're going to do that dephosphorylation inside of the ER. So this glucose 6-phosphatase has its kind of enzymatic site in the lumen or the inside of the ER. So this glucose 6-phosphate has this transporter in your liver that takes it into the ER. In the ER, the phosphate group gets removed, we get free glucose. That glucose can then get shipped out of your liver and into your bloodstream. By kind of keeping it hidden like this, we prevent a fetal cycle of phosphorylation and dephosphorylation, glycolysis and gluconeogenesis happening at the same time. Okay, so that's the key thing about how these enzymes actually work, and now we'll get into their regulation. But there are a few key things that I wanted to say. One is that there are a lot of different glycogen storage disorders that are associated with various mutations in all sorts of these different enzymes of the pathway. It's not that you're responsible for knowing these things, just that I know that a lot of you are interested in medicine. And so we talk about like glycogen storage disorders, things like Pompe disease, McArdle disease, Hirsch's disease. There are various different glycogen storage disorders that often affect the liver, the skeletal muscle. Makes sense because that's where we have a lot of our glycogen metabolism going on. And if you have problems with the enzymes of glycogen metabolism, you can have very large problems. And so some of them are more minor, some of them are more serious, but that is something that you might encounter if you go in the medical direction. There's going to be a lot of hormonal regulation. So I would also like you to put tabs in your spreadsheets for the various hormones that we're going to encounter. So, and fill these in as we go. So you'll want to tab for insulin, glucagon, epinephrine. We're not really going to talk about cortisol yet, but then for each of these tabs, you're going to have multiple like sub-tabs, and this should make more sense as we go along. The steps that are highly regulated are going to be these bolded things here. So we're going to see our glycogen synthase and our glycogen phosphorylase. They're going to be regulated in a way that makes sense. So although the actual mechanism of the regulation, like the details and stuff, is going to be kind of complicated, the logic of it is, well, fairly simple. You need to think about, well, what's the goal? You don't want to build glycogen, so do glycogenesis and break it down so that is glycogenolysis at the same time. So we're going to regulate them reciprocally. We can have like one hormone activate a signaling pathway that activates a kinase. That's going to activate one enzyme, but inactivate another enzyme. So it can activate one that breaks down glycogen and inactivate one that builds glycogen, and vice versa. We're going to see a very similar concept when we talk about the regulation of glycolysis and gluconeogenesis. You don't want to be breaking down sugar at the same time you're making sugar. That's just going to be a futile cycle, wasting energy. Another core concept is that, well, if blood sugar is low, you want the liver to break down glycogen and ship out glucose. This is going to raise your blood sugar to normal. And remember, what's the signal for when our blood sugar is low? What's the hormone we use? Right, our glucagon. Glucagon says, hey, the glucose is gone. Let's ship some out. Conversely, if blood sugar is high, well, now you want the liver to take it in and you want it to store it as glycogen. This will lower your blood sugar to normal. What's the hormone that signals that? Right, that's our insulin. The insulin says, let the glucose in, and that will lower our blood sugar. That's in terms of what the liver is doing, but cells need energy too, and they can store glycogen. So the liver is like the main storage source for glycogen. It could be like 10% of your liver weight or something can be glycogen. Your muscles can also store glycogen, but they store it for a very different reason. So remember, your liver is like regulating your blood glucose, but your muscles, they want the ATP for themselves. So they can break down the glycogen, get glucose that they can then use for themselves to take through glycolysis to make ATP. As we'll see, and remember that the liver can do gluconeogenesis, and it can do that last step of gluconeogenesis, which actually takes that glucose 6-phosphate and frees it into free glucose that can release. Your other cells, like your muscle cells, they don't have that glucose 6-phosphate. So even if they were to make glucose 6-phosphate, they couldn't ship it out. And so they get to keep it for themselves. Okay, so a little more detail. What are we going to look for when we want to upregulate glycogenesis and downregulate glycogenolysis? So remember, genesis, this is the making. So we want to make glycogen, that is, piece together glucose to make glycogen, and downregulate glycogenolysis. So we want to stop breaking it down. We'd want to do this if we already have high glucose in our bloodstream. Signals of this would be, well, glucose 6-phosphate, and insulin. We also don't need to break down, we also don't need to free up glucose if we have enough energy. So if we have a bunch of ATP, then let's not do that. Let's instead store that glucose. If, however, we have low glucose, well, then what we're going to have is we're going to have our glucagon signaling. We're going to have signals of low energy. We're going to have a higher concentration of AMP and ADP that can act as allosteric regulators. And we'll see in our muscles that, well, if they need an energy boost, when muscles contract, they have calcium signaling. And this is going to help promote the effects of adrenaline, the effects of our pKa, and things like this. Again, we'll go into the details, but if you think about kind of the big picture, you can logic your way through things. Okay, so we already talked about how we're going to see covalent regulation and allosteric regulation, and you should be able to identify what type of regulation is going on and give examples of each. The regulatory enzymes that we're going to see our pro-glycogenesis or anti-glycogenolysis enzymes. These are going to be PKB, AKT. So remember, that's going to be one of those kinases that was stimulated by insulin. So when we talked about insulin, we focused a lot on like the RAS, RAF, MEK, ERK pathway, but now we're going to go and we'll look back more at that other pathway. This pathway that went through our PI3K and activated PKB. So remember, we still start with IRS1, but here we're activating the kinase PA3K. That's going to take PIP2, make PIP3. That's going to activate PKB. PKB is also known as AKT, so you'll see it in both ways. And this is then going to have effects downstream on our glycogen metabolism. We'll also see that insulin is going to stimulate a phosphatase called PP1. This is our phosphoprotein phosphatase. And we'll look more in detail about how it's going to do that. And the other side of things, if we want to inhibit glycogenesis and promote glycogenolysis, so promote the breakdown and inhibit the linking up. Here, we want the opposite. we're going to want like our glucagon signaling. And this is going to signal largely through our pKa or CAMP-dependent protein kinase. So remember that our beta-adrenergic receptor, as well as like an epinephrine receptor. So your liver has glucagon receptors, but your muscle cells and other cells don't really have that glucagon receptor, other than like because they can't do gluconeogenesis, so it doesn't make sense for them to respond to glucagon. But they do have to respond to other stimuli, such as the need, like that fight or flight response to your adrenaline. So these can both feed into similar G protein pathways that can activate your pKa, your protein kinase A. This is sometimes called cyclic AMP-dependent protein kinase. Those are then going to have pleiotropic effects. If you're not familiar with this word, pleiotropic, that would be a great one for your glossary. Basically, it means that it has a lot of different effects. So it doesn't just do one thing, it does multiple things. Basically, it's complicated, is what that pleiotropic term is indicating. So we'll see how this pKa is then going to stimulate phosphorylase kinase, and it's that's going to further like amplify the signal. And then we'll also see that we're going to have this kinase GSK3, this glycogen synthase kinase. So as we go through, you want to keep your eye out for these enzymes, PKB, PP1, pKa, phosphorylase kinase, and GSK3. These are going to be our key regulatory enzymes in our pathway. And then the ones that we mentioned before were the metabolic enzymes. Okay, I know this is a lot. So let's just kind of put things back into perspective, and then we'll go into the details. So glucagon, we say all the insulin, all the glucose is gone. Glucagon is secreted by the pancreatic alpha cells in response to low blood sugar. It's going to stimulate our catabolic processes. So we break things down for energy or for glucose that we can ship out in our liver. And it acts largely through pKa, so this CAMP-dependent protein kinase signaling. Insulin says better let some glucose in, so we have high blood glucose. Our pancreatic beta cells are going to ship out insulin. It's going to stimulate anabolic processes. So we've got a lot of glucose, let's do stuff with it. Our liver is going to take it in and store it as glycogen, and you can convert it into other things. And this is the effects of insulin are largely mediated through PKB or AKT signaling. I keep harping back to this main picture because really keeping things in this perspective is really going to help you logic things through. So don't try to memorize, just try to kind of like logic your way through things. And really you just have to kind of like look at these pathways a lot. I'm going to show diagrams in various different forms. Some of them might make more sense to you than others. So just kind of find what works for you. Really, like a lot of it is just stuff that you kind of have to sit with. You kind of have to think about over and over. It's not stuff that you can easily learn in a quick session. So don't feel bad if this video takes you a while to go through, if you need to keep pausing and returning. I've literally read this section of the book so many times and gotten more out of it each time. And so I don't want you like spending a ton of time when you have all sorts of other stuff to do. But if you really want to understand the pathway, it's going to take some time. That being said, here's the big picture overview. So remember, we have glycogen. We can break it down to release glucose 1-phosphate that then we can interconvert through our phosphoglucometase to make our glucose 6-phosphate. That glucose 6-phosphate, we can then dephosphorylate it in our liver to ship it out. In our muscles or our other cells, we can break down the glucose 6-phosphate and use it for energy. We could also take that glucose 6-phosphate and we can do other fates for it. We could go and we could take it through the pentose phosphate pathway. We could take it down through the hexosamine pathway. So various fates for that glucose 6-phosphate that we could get from this glucose 1-phosphate that we could get from our glycogen. The breakdown of glycogen is coming from this glycogen phosphorylase. Conversely, we can make glycogen by piecing together glucose 1-phosphate from glycogen synthase. Okay, so now we want to regulate this glycogen phosphorylase and the glycogen synthase. They're going to be reciprocally regulated. We're going to see that much of this regulation is through the phosphorylation by pKa. So remember, that was stimulated by our glucagon and our epinephrine, and countered by dephosphorylation by PP1, which was stimulated by insulin. Some of these enzymes are going to be active when they're phosphorylated, and some are going to be inactive when they're phosphorylated. And the same kinase is going to be able to phosphorylate them all, and the same phosphatase is going to be able to dephosphorylate them all. Hence, we're going to be able to have this reciprocal regulation. Glycogen phosphorylase. So remember, this breaks down glycogen to give us our glucose 1-phosphate units. It's going to be active when phosphorylated, and it's going to be phosphorylated by phosphorylase kinase. Phosphorylase kinase is going to be activated when it's phosphorylated. And these can be phosphorylated by our pKa, which remember was stimulated by glucagon and epinephrine. Conversely, when we have glycogen synthase phosphorylated, well, now it's inactivated. And if it got phosphorylated by our pKa, so pKa is therefore able to inhibit the glycogen synthase. Our PP1, our phosphatase, is able to reverse the action of all of that, activate our glycogen synthase, and inactivate our glycogen phosphorylase and our phosphorylase kinase. That's the big picture, and then we have multiple layers of regulation that go on top of it. Let's start by reviewing the key things that we want to keep in mind when we consider the details of how they're regulated. So our glucagon and our epinephrine. So glucagon, this signals low sugar. Our epinephrine, this is our fight or flight response. Both of them are going to stimulate glycogenolysis, so free up glucose to either ship out in your liver or to use to break down in glycolysis in order to get energy to fight or flight. And you don't want to be building up glycogen, so we're going to inhibit glycogenesis. Insulin, this is the opposite. Here we have our high blood sugar. We basically want our liver to take that sugar in and piece it up together to form glycogen. We don't want it to break down more glycogen because that would then make the situation worse. So we'll inhibit glycogenolysis and stimulate glycogenesis. As for the how, glucagon and epinephrine, they're going to stimulate the phosphorylation and thereby the activation of glycogen kinase and glycogen phosphorylase. So glycogen kinase and glycogen phosphorylase are both active when they're phosphorylated, and they're going to be phosphorylated in response to glucagon and epinephrine. On the other side of things, we have that glycogen synthase is going to be inactivated by phosphorylation, and that phosphorylation is going to be promoted by our glucagon and our epinephrine. Insulin is going to do the exact opposite. Through the actions of PP1 largely, it's going to promote the dephosphorylation of those enzymes. That's going to reverse the effects, so our glycogen kinase and our glycogen phosphorylase are going to be inactivated, and our glycogen synthase is going to be activated. That in mind, let's get into the how now. So glycogen phosphorylase, remember, what does it do? Right, it's going to break down our glycogen. So we want it to be active in response to glucagon and inactive in response to insulin. It's not just glucagon that we're going to want to respond to, we're also going to want to respond to epinephrine. So glucagon, remember, we're breaking sugar down to release it. And epinephrine, we have our fight or flight response. Yes, we want to give glucose to our other tissues, but then within individual tissues, we want to stimulate glycolysis and stuff. So we want to break down that glycogen, get glucose that we can use for ourselves. And we'll see how these different pathways work similarly and also kind of have differences. So we are able to have differences in part because we can have different isozymes or versions of the enzyme in different cell types. We're not going to worry too much about the distinctions between the liver and the muscle versions of these enzymes, just about kind of knowing what regulates them. One tricky thing is that people give these different names to the phosphorylated and the non-phosphorylated form. So phosphorylase A, this is the active form. So when you see A, think active. Whereas phosphorylase B, this is the less active form. In the case of our phosphorylase, here our phosphorylase A, the active form, is going to be phosphorylated. So it's phosphorylated in the active form, dephosphorylated inactive form, phosphorylated A form, dephosphorylated B form. So how does it get phosphorylated? It gets phosphorylated by phosphorylase kinase. And phosphorylase kinase is activated when it's phosphorylated. Phosphorylase kinase is phosphorylated by pKa, and pKa is stimulated by glucagon and epinephrine signaling. So basically, we have a situation like this. We have our cyclic AMP, we have our G protein coupled receptor is going to, through its receptor process, then go and it's going to stimulate adenylyl cyclase. Adenylyl cyclase is going to convert ATP to our second messenger, CAMP. CAMP is then going to bind to pKa and activate it. pKa is then going to phosphorylate our phosphorylase kinase. Our phosphorylase kinase is then going to phosphorylate and activate our glycogen phosphorylase, or what we commonly refer to as just phosphorylase. So in this way, we're able to have this cascade. Another thing though, so this was happening basically through this pathway that we talked about. We also talked about how there was a different pathway from our G protein coupled receptors, where we went through we could go through this phospholipase C pathway where we generated this IP3 and DAG, and the IP3 stimulated the calcium release. The calcium release then stimulated these calcium binding protein called calmodulin, and then other proteins that bound to calmodulin got activated. And so we can have calcium be released in response to these signals from like our glucagon or our epinephrine. This is going to serve as like an allosteric regulator that's going to help get this process going and stimulate it further in our myocytes. So in our muscle cells that don't have those glucagon receptors, but do have those epinephrine receptors. So remember, both of these receptors are able to stimulate the pKa. But in addition, so you don't just want it to be like when you have a ton of adrenaline that you need your muscles to break down glucose or glycogen. You also want it kind of just when your muscles are working really hard. One of the signals for muscle contraction is going to be calcium release. So we get calcium release in our muscles when they're working, as well as we get calcium release through that pathway we were just looking at. So both of these ways we're able to kind of get increased calcium, and that calcium is then going to stimulate phosphorylase kinase allosterically. So remember, with allosteric regulation, we're binding to a site other than like the active site, and we're causing the protein to kind of like change shape and become more active. In this case, what's going to happen is basically we have these regulatory sites where in order to get full activation of this phosphorylase kinase, you need both the phosphorylation and the calcium binding. So that's going to further stimulate this effect. Another signal that you want to break down glycogen, especially in your muscle cells, is going to be AMP. So if you got low energy, then you're going to want to stimulate more breakdown of glycogen so you can get more glucose so that you can use for yourself. AMP is going to basically accumulate in your active muscle. It's going to bind to and activate the phosphorylase. So you have another regulatory site where we can basically have this AMP bind and further stimulate things. On the other hand of things, if you've got a lot of ATP, you don't want to break down more glycogen, you don't need more glucose. Here, ATP is going to block that allosteric site that the AMP normally bound to, inactivating the phosphorylase. So in the end of the day, what we have is that this phosphorylase is going to be activated when we've got glucagon and epinephrine signaling, when we've got our calcium and our AMP high. And it's going to be inactivated when we've got the ATP high, whereas we'll see when we have this phosphatase activated by our insulin signaling. So glycogen phosphorylase is active when it's phosphorylated. It's phosphorylated by phosphorylase kinase. Phosphorylase kinase is also active when it's phosphorylated. Both are inactivated by dephosphorylation. And that dephosphorylation happens from PP1, or phosphoprotein phosphatase 1. PP1 is going to be activated by insulin signaling. As to the how, you don't really need to know too much the details of how PP1 is activated by insulin, just that it is activated by insulin. I'll just tell you so that you know. Basically, what happens is that there's that pathway of insulin signaling that goes through PI3K and PKB. The insulin receptor is going to, when it's activated, it'll phosphorylate IRS1, which will activate PI3K, which will activate PKB. PKB, well, what it can do is it can then go and it can actually phosphorylate a regulatory subunit of PP1, which is going to activate it. Now, PP1 can then go dephosphorylate and activate glycogen synthase, as we'll talk about, Or what we just mentioned, it can dephosphorylate and inactivate phosphorylase kinase and phosphorylase. So it can directly dephosphorylate both the phosphorylate kinase and the phosphorylase. As a result of both of them, so if you inactivate the kinase, you get less of this phosphorylation, and you get less of this phosphorylation too if you basically just remove it. So both of those are going to have the effect of making it so that you break down less of your glycogen. So that was the phosphorylation. So that was the phosphorylation by PKB that was going to basically activate PP1 indirectly. We have a different situation with pKa. So remember, you can think of A, the adrenaline, think of A, the alpha cells, the glucagon, and B, this is going to be the beta cells, your insulin. And so it's not really that's just happening in beta cells or alpha cells, instead we're talking about like our muscle and our liver. It's just kind of like a mnemonic to help you remember that PKB goes with insulin and pKa goes with glucagon and epinephrine. So in the case of our glucagon, well, what do we want to do here? We want to break down glycogen, we want to do glycogenolysis and inhibit glycogenesis. pKa is going to phosphorylate and activate an inhibitor protein of PP1. And basically that's going to, then this phosphorylated inhibitor is going to bind to and inactivate PP1. So this is going to basically cause PP1, the phosphorylation causes PP1 to kind of dissociate too from this glycogen, and so make it so that it can't do the dephosphorylation. So everything's kind of like held together. The phosphorylation by pKa is going to make it so that the PP1 is inactive. The phosphorylation by the pKb is going to make it so that it's active. You don't need to worry about the details of this. Just know that insulin is going to stimulate the activation of PP1, and that our glucagon and our epinephrine are going to stimulate the inactivation of PP1. I just wanted to show you how, because I think it's cool, and I would want to know. I'll talk a little bit more about allosteric regulation. So that was our hormonal regulation for the most part. We talked a little bit about allosteric regulation in terms of our like ATP and ADP. Another sign of energy status is going to be our glucose and our glucose 6-phosphate. We'll see that, well, if you got a little glucose, what are you going to want to do? You're going to want to build it up together. You're not going to want to make more glucose by breaking down glycogen. So basically, we want our glucose and our glucose 6-phosphate to inhibit glycogenolysis, because we got plenty of glucose already, and activate glycogenesis. So this inhibition would be feedback inhibition, and this activation would be kind of like feed-forward regulation. This is going to happen through our glucose 6-phosphate and our glucose acting on different molecules. Glucose 6-phosphate is going to allosterically activate PP1. So that phosphatase that we just saw, remember that phosphatase was going to dephosphorylate and thus activate our glycogen synthase, and it's going to dephosphorylate and thus inactivate our glycogen phosphorylase and our phosphorylase kinase. It binds to PP1 and activates it. It also makes it easier for PP1 to do its job. Glucose is going to bind to allosteric regulatory sites on our phosphorylase. This is going to make it so you get a conformational change that kind of pops out those phosphate groups into a form that's more accessible for PP1, making it easier for PP1 to do its job. So you have this kind of combined regulation where the glucose and the PP1 are able to act together to kind of amplify the signal and make it so that you can have kind of like a safety check that you really do want. You really do want to turn the phosphorylation off. Okay, so that was our phosphorylase. Now let's look a minute at the synthase. So our glycogen synthase, this is kind of like the opposite in terms of when it's active and when it's inactive. So our glycogen synthase is going to be inactive when it's phosphorylated. So just like we talked about how our glycogen, how our phosphorylase had two forms, and we called the active form the A form. Similarly, our glycogen synthase has two forms, and the active form is the A form. But here, the active form is unphosphorylated and active, and the B form is phosphorylated and inactive. The phosphorylation is happening by the same pKa protein, as well as by a protein called GSK3 or glycogen synthase kinase 3. So both of these are able to phosphorylate glycogen synthase A to give you glycogen synthase B. Remember that A and B just refer to like the forms of it, like whether or not it's phosphorylated or not, but it's the exact same protein, just modified or unmodified, active or inactive. So I find the kind of like A and B notation to be super duper confusing, but you will see it used quite often. The key thing is that when it's phosphorylated, it's inactive. When it's dephosphorylated, it's activated. It's phosphorylated by pKa, which remember was stimulated by our epinephrine and our glucagon signaling, and it's by GSK3. And it's dephosphorylated by that PP1, which was stimulated by our insulin signaling. A note about GSK3. Well, GSK3 is going to be inhibited by our insulin. So basically, we'll have that PKB that got stimulated by insulin. It's actually going to phosphorylate and inhibit GSK3, keep GSK3 from doing its job of phosphorylating and thus inactivating our glycogen synthase. I don't need you to worry about the details. What I would like you to know is that insulin is going to inhibit GSK3. So it's indirect, but it does inhibit GSK3 and stimulate PP1. Both of those are going to have the effect of dephosphorylating glycogen synthase, either directly promoting the dephosphorylation or preventing the phosphorylation by GSK3. I will also like you to know that pKa is able to do this phosphorylation, and pKa is that one we're more familiar with, that kinase that we see with our glucagon in our epinephrine signaling. Those are both going to have the effect of inactivating glycogen synthase, but then all of that can be reversed by our PP1, our phosphoprotein phosphatase 1, which was stimulated by our insulin. We also, you'll also see that we have that glucose-6-phosphate is also able to allosterically regulate glycogen synthase. So just how we saw that glucose was going to kind of make it easier for PP1 to dephosphorylate our phosphorylase kinase, it's also glucose-6-phosphate is going to make it easier for PP1 to dephosphorylate and activate glycogen synthase. So glucose and glucose-6-phosphate have these kind of like similar roles that help make PP1's job easier. Glucose is going to make it easier for PP1 to dephosphorylate our phosphorylase kinase, and glucose-6-phosphate is going to do similarly for glycogen synthase. So both these are kind of signaling that we've got tons of glucose building up. And so let's piece it together. Let's not break down more glycogen. So let's look at the big picture and see what we've seen and where we're going. So in terms of what's happening in the liver, in response to high blood glucose, you'd want your liver to basically take the glucose in, piece it up into glycogen, store it and for later, and reduce your blood sugar. The signal that our pancreas sends out in response to high blood glucose is insulin. That's sent out by our beta cells of our pancreas. That's going to stimulate various insulin-sensitive protein kinases. I found this kind of a little confusing, but from what I could tell, basically this is just talking about like PKB, as well as other insulin-sensitive kinases. These are going to have the effect of stimulating our PP1. PP1, that's that protein phosphatase. It's going to then dephosphorylate phosphorylase kinase, reduce phosphorylase kinase activity. It's going to dephosphorylate glycogen phosphorylase and reduce glycogen phosphorylase activity. It's going to dephosphorylate glycogen synthase and activate that glycogen synthase activity. As a result of inhibiting this phosphorylase kinase and the phosphorylase, you're inhibiting glycogen breakdown. And by activating glycogen synthase, you're activating glycogen synthesis. PKB is also able to phosphorylate and inhibit GSK3. And what that is going to do is that's going to prevent GSK3 from inhibiting glycogen synthase. That's going to further stimulate glycogen synthesis. Next, we're going to look at kind of our regulation of glycolysis and gluconeogenesis. We'll see that the high blood glucose is also going to kind of make the cells synthesize more hexokinase 2, PFK1, and pyruvate kinase. So basically, it's going to do more of glycolysis in your liver. When you have this, when you have a lot of glucose, you're going to basically want to use some of the glucose. On the other side of things, what if we have low blood glucose? Well, in this case, we're basically going to want our liver to break down glycogen and send out glucose. So in response to low blood glucose, now your pancreas, the alpha cells this time, are going to send out glucagon. Glucagon is then going to signal through the cyclic AMP and activate pKa. pKa is going to phosphorylate our phosphorylase kinase. Phosphorylase kinase is when it's phosphorylated, it's activated. It's going to phosphorylate and activate glycogen phosphorylase, and that's going to then go and break down glycogen. pKa is also going to phosphorylate and inactivate glycogen synthase, and that is going to decrease glycogen synthesis because glycogen synthase, when it's phosphorylated, it's inactivated. So again, we have our reciprocal regulation. When we look at regulation of glycolysis in response, well, here we're going to basically decrease the expression of decrease the activity of PFK2 and increase FBPase2. Basically, what we're doing is we're inhibiting glycolytic enzymes and we're activating our gluconeogenic enzymes. In response, we're going to have decreased glycolysis and increased gluconeogenesis, so we can make more glucose from scratch, in addition from getting it from the breakdown of our glycogen. So let's just review these different steps that we've seen. Insulin inhibits glycogenolysis and stimulates glycogenesis. This is through PKB, also known as AKT, and or other insulin-sensitive kinase signaling. So through the PP1, our PKB is going to phosphorylate the regulatory subunit of PP1, activate it, and basically then our PP1, once activated, is going to dephosphorylate and thereby activate our glycogen synthase, so we make more glycogen. PP1 is going to dephosphorylate and thereby inactivate phosphorylase kinase, and so that we have less phosphorylase activated. PP1 is also going to dephosphorylate and inactivate phosphorylase, thereby less glycogen is broken down, so either directly or indirectly. Now we have through PKB or AKT, we're also going to stimulate this glycogenesis. PKB is going to phosphorylate and inactivate GSK3, so our glycogen synthase kinase. That's going to prevent GSK3 from phosphorylating and inactivating glycogen synthase. As a result, more glycogen is made. Now let's look at the other side of things, glucagon and epinephrine. So remember glucagon, we've got low blood sugar, our glucose is gone. Epinephrine, we want our fight or flight response. Both of these are going to stimulate glycogenolysis, free up some glucose, and inhibit glycogenesis. Stop storing the glucose. Let's use it, or let's ship it out. It's going to act through pKa signaling. pKa is going to phosphorylate and activate phosphorylase kinase. Phosphorylase kinase, in turn, is going to phosphorylate and activate phosphorylase. And then that active phosphorylase is going to break down glycogen. pKa is then going to also going to phosphorylate and inactivate glycogen synthase. As a result, less glycogen is made. pKa is also going to phosphorylate and activate PP1 inhibitory protein. If you activate the inhibitor protein, it prevents the dephosphorylation and thus the inactivation of phosphorylase, and it prevents the dephosphorylation and thus activation of glycogen synthase. So yes, the details can get complicated, but think about the logic. Think about when you want to upregulate glycogenesis and downregulate glycogenolysis. So those would be signals like high glucose, high energy, so where like our ATP levels are high. And conversely, you want to downregulate glycogenesis and upregulate glycogenolysis if you've got low blood glucose. If you've got low energy, so like our AMP or ADP levels are high, or if your muscles are working hard and you've got that kind of like calcium release. All of those are going to make it so that you can break down glycogen either for your own use or to basically send to cells throughout the body, in the case of our liver, which had that G6Pase so that it could actually dephosphorylate the glucose 6-phosphate and let it out.